Hello, this is the fifth lesson in the series on cell biology on stem cells. The lesson objectives are to describe what stem cells are, to compare embryonic and adult stem cells, to describe the importance of stem cells. This lesson is tailored to the AQA, OCR, and Edexcel GCSE specifications and any other secondary specification. Let's start by looking at what stem cells are. Stem cells are undifferentiated cells that can develop into any other type of cell. There are two types of stem cells, the embryonic stem cells obtained from the embryos and the adult stem cells which can be obtained from more or less any part of the body, but the common place is the bone marrow. Embryonic stem cells are unspecialized cells obtained from the embryo which can grow into any type of cell. The adult stem cells are specialized cells found throughout the body and can only develop into the same type of cell. Undifferentiated stem cells in plants are known as meristems. Meristems are found in the shoot and roots of plants and differentiate throughout the plant's life. Let's look at the uses of stem cells. They can be used to replace cells that have been damaged or destroyed, for example, in type 1 diabetes, in cases of multiple sclerosis which can lead to paralysis, and in cases of spinal cord or brain injury which have led to paralysis. They are also important in medical research such as on how cells differentiate and in the testing of drugs. Now let's look at therapeutic cloning. Therapeutic cloning is a process of creating stem cells with the same genes as the patient. Because therapeutic cloning uses genes from the patient's own body cells to create new organs, there is less chance of the immune system rejecting it. It is important in creating vital tissues or organs that can treat kidney and other disorders. Now let's look at the stages in therapeutic cloning. So in stage 1, step 1, the nucleus is taken out of the human egg cell, and the nucleus from the patient's body cell is put into the empty human egg cell. The egg cell is stimulated with electric shock to develop into an embryo. The stem cells are extracted from the embryo after 4 to 5 days, and the stem cells are cultured in a petri dish. They are then treated to develop into desired specialized cells. Now let's look at some of the issues associated with stem cell technology. So we look at the disadvantages, the social issues, and the ethical issues. If the stem cell is contaminated by virus, it could be transmitted to the patient, which could have negative implications. It is not 100% efficient, as cultured stem cells could become mutated and develop into a tumor. There is low number of stem cell donors. In terms of the social issues, the question is whether the public is being educated about what stem cells are and can and cannot do. Do the benefits of stem cell technology outweigh the risk? Are patients being exploited by paying for expensive treatments and being given false hope of a cure, as stem cell therapies are only in their developmental stages? The ethical issues are: what stage an embryo can be regarded as a person? Is it right to create embryos for therapy and in the process destroy it? Could an embryo be a resource for researchers? British law now allows that embryos can be created for scientific purposes. Now let's look at this true or false plenary to summarize the lesson. Stem cells are specialized cells. That is false. They are undifferentiated cells. Embryonic stem cells can develop into any type of cell. That is true. Stem cells in plants are known as meristems. That is true. Adult stem cells can be obtained from the bone marrow. That is true. British laws allow stem cells to be used to make designer babies. That is false. In therapeutic cloning, stem cells are created using genes from a donor. That is false. It's genes from the patient. Stem cells are used in treating type 1 diabetes. That is true. In therapeutic cloning, chemicals are used to stimulate the egg cell to divide. It's not chemicals, it is electric shock. An ethical issue with stem cells is whether it is right to destroy embryos for stem cell technology. That is true. Stem cell technology is 100% efficient. That is false. See you in the